Dear students, we'll be now starting the chapter on concept of gene mapping. That means we'll be measuring the distance between two genes based on the experiments done on Mendelian genetics. So before starting that exercise, let's just run through the basic concept of linkage. How we can assess whether a gene is linked or not. That means alleles of different genes present in a chromosome could be linked or could be independent. When we say it is linked gene, that means the two alleles present side by side in two different loci are passing from one to the next generation as it is in the same conformation. In the same conformation. If it is not, then we say genes are not linked with each other. In this picture, the independent assortment, two genes on two different homologous pair of chromosomes are shown. In one side, there is gene A and there are two forms, capital A and small a. Gene B has two forms, capital B, small b. So this capital A and small a are the alleles of each other. Capital B and small b are the alleles of each other. And these alleles are present in two different chromosomes, homologous chromosome. These are the homologous chromosomes. Okay. So one pair of the homologous chromosome is orange in color and another pair in blue in color. So let's suppose A gene is present in chromosome number 1 and B gene is present in chromosome number 2. So as per Mendel's law, second law of independent assortment, when gametes are formed, the alleles will be segregated independently. Okay, so in one of the gamete, you can see that left panel. So there are four circles. There are four circles you can see. So in left upper circle, you can see gene A and gene B are going together because these are the two different chromosomes. So they are now in haploid conformation. So only one strand of A and one strand of B is there, not both. Okay, because now it's a homologous. Now it is not in homologous pair. It's in haploid pair. So in the upper panel, we can see a bivalent chromosome and chromatids are shown. Chromatids are shown, right? So in this case, it's not chromatid, it's a compact chromosome. So there are two chromosomes, one harboring allele A, another harboring allele B. Both are dominant in this case, or wild type you can say, both are capital. Left hand side below panel, lower panel, there is small a allele and capital B allele. Right hand side panel, upper side, capital A, small b, and lower panel, right hand side, small a, small b. These are now independently assorted. Okay, so there is no, you can say nothing like a linkage concept in this case. It's independently assorted, and that's how gametes are formed. So you have to understand this particular concept while we'll be doing analysis for linkage mapping. So in the upper panel, we can see chromatid-wise distribution of alleles in two homologous chromosomes, and in lower panel, gametes are shown in haploid conformation. Now, if you look at this picture, in left hand side panel, you can see in a same chromosome, in a same DNA strand, capital A and capital B are present. left hand side panel, image number B. So in orange chromosome there are no two arms, so it's a you can say chromatids, two chromatids. So capital A and capital B, two different genes are present. So therefore two different genes you can see marked in black. So these are the two different loci harboring two different genes, and each gene are having two alleles, capital A and small a, capital B and small b. In the when gametes are formed from this one homologous chromosome pair, so one allele, one gamete is carrying capital A, capital B, another small a, small b. Obviously, there is no crossover. There is no crossover, there is no exchange between chromosomes or non-sister chromatids because these are homologous chromosomes. So, as it is whatever seen in parental chromosome, so upper panel is parental chromosome and this is the gamete of the parental chromosomes. In the right hand side panel, the same kind of conformation is shown, capital A and capital B, small a and small b, but two non-sister chromatids are now crossover. Okay, so non-sister chromatids are crossed over and they have exchanged a part of their chromosome. So in picture number C, left hand panel, you can see A and B allele in non-crossover gamete. So when gametes are formed from non-crossover chromosome, non-crossover you can say chromatid, you can see both A and capital B is present as it is. So you can see the upper panel where crossover is shown. The upper chromosome, the uppermost chromatid is having capital A and capital B. So first gamete is capital A, capital B as it is, and it is non-crossover. Lower panel, you can say crossover gamete, where capital small a is now linked with capital B after crossing over. You can see the blue chromosome upper panel. Capital B is now added with small a. This is happening because there is a crossover. So now this is a crossover gamete. So it is now, the gene order is now rearranged. Now small b is not coming with small a. Capital B is coming with small a. Okay, the same way right hand side, capital A is now coming with small b because of crossover. So after this one crossover event between two non-sister chromatids, now you can see two crossover gamete and two non-crossover gamete. So this is the basic event and basic rearrangement happens when there is one single crossover between two non-sister chromatids. And when alleles of two different genes are present on two different sides of the crossover site. Crossover site you can identify, you can see the X structure, and two alleles, small a or capital A and small b or capital B, are present at two opposite sides of that crossover site. So that is why you can see this rearrangement. So in this picture, we'll just go through the complete linkage structure, linkage event that happens with an example of actual gene. So these HV and BW are the two genes governing eye color and the wing vein structure of Drosophila fly, fruit fly. Okay, so in this case you can see in parental conformation P1, these two genes are present in same chromosome and side by side, two black bands, HV and BW. So HV plus means it's a wild type, BW plus means it's wild type. Wild type means they are showing the wild type feature. HV plus is a red color eye, HV is a white color eye, BW plus is having thick venation vein, and BW is having thinner vein. So these are the phenotypes: brown eye and thin vein, and is crossed with heavy veins and red eye. Okay, so these are the conformation. When gametes are formed, you can see straight away, example wise, one gamete is having HB plus BW, orange one, and in the right hand side a blue gamete is formed, just to show without any crossover, because there is no crossing over, so there is no exchange between homologous pair of the chromosome. And you can see a two parental type chromatids are now came into the gamete. Now in F1 generation, when two gametes are met, now you can see a thin vein red eye. Thin vein red eye. So thin vein red eye F1 generation individual cannot be traced back to the parental type. Okay, because parental type are different. You can see the phenotype. So this F1 phenotype is a new phenotype because of simple zygote formation from two different parent. This is not an event of recombination. It's just the event of, you can say, just reproduction. Just two gametes are fused and zygote is formed. So therefore the phenotype from two different parents are now unified. And now when it is test crossed with obviously recessive or the not mutant type gametes, mutant type individual with heavy veins and brown eye, the gametes are formed. So because of complete linkage, F1 individuals form only parental gamete. Okay, so F1 individual only can form parental gamete. Parental gamete means gametes are same as the parental gamete, just above that, within that oval shaped circles. You can see the gametes are same from F1 because there is no linkage. If there is any linkage present, the F1 gametes will not be same as the parental gamete. Okay, there will be a rearrangement, but in this case there is no rearrangement. Gametes are same as parental gamete. Now from test cross, now that recessive individual, gamete is now formed. Okay, so these are the gamete conformation after F1 generation when there is no linkage. I mean, when there is a complete linkage, when there is no crossover. So when you already saw, you have seen this kind of 2x2 table where the different gametes formed in this picture is now arranged in two opposite side, and these are the progeny. So when F1 is now selfed with F1, so F1 are having two alleles, this allele HB plus and BW and HV BW. So when they are crossed selfed, so these four different type of individuals, four different type of progeny can be found. 1/4 thin and brown, 2/4 thin red, 1/4 heavy red. So the ratio is 1:2:1. Okay, and when it is test crossed, F1 is test crossed, the ratio will be 1:1. Thin brown and heavy red, the ratio will be 1:1. Okay, so the main essence of this, why I'm saying is that if you look at this F1 and test cross progenies in that B picture B, the progeny after this crossover, this crossing, the progeny are same as the parental type: thin brown, heavy red. Let's see this thin brown, heavy red. Okay, so these progeny are same as the parental type P1. So this thing is possible when there is no linkage. when there is complete linkage, when there is no crossover, I mean to say. Let's see what happens when there is a crossing over. When there will be crossing over, always a mutant type allele will be formed. Mutant type allele. So in that panel A and panel B, you can see in panel A there is no crossover. So the blue chromosomes and green chromosomes are not crossing over with each other. So the alleles a small b and plus alleles are present on the same chromosome pair, and as it is coming in the gametes. So the result is 4 non-recombinant chromatids, okay, from two homologous pair of the chromosome. And when there is a crossing over event happens, so in that right hand side panel, picture B, crossing over between these chromatids. So these are the non-sister chromatids, and chromosome arms are being crossed over, and now there is a rearrangement. Now there is rearrangement. A is coming with plus allele, and B is coming with plus allele. So you can see there is a hybrid formation, blue and green. And when gametes are formed, two recombinant and two non-recombinant chromatids can be seen. Because why there is two recombinant? Because two chromatids are now exchanged part of their chromosome with each other. So the alleles are now exchanged between two non-sister chromatids. So when many times there is crossover, but there is no recombination. There is no recombinant happens. Why? Because crossing over is happening outside of these two alleles. Okay, outside of these two alleles. So now this particular chromosome at the right hand side, the blue chromosome is fused. Part of the chromosome is fused with the green. Okay, but alleles are not rearranged. Still, small a is coming with small b, and capital A is coming with capital A. Capital B. Okay, because the crossover event is happening outside of these two genes A and B. In the lower panel again, there is two crossover site, right? There is two crossover site, but again the alleles are not exchanged, though chromosomes are now hybrid, but these are non-recombinant. Because why it is not recombinant? Because there is no recombination. There is rearrangement of the pair of the chromosome segment of the DNA, but still alleles are arranged in same order. Unlike in the previous slide that I have shown, that plus and A and plus and B are now exchanged with each other. In this present picture, still capital A and capital B and small a and small b are present in same chromosome, same same conformation. So there is no recombination happening, though there is crossing over. So you have to understand these different concepts, that crossover will never give you recombinant. I mean to say, not always. It will only give you recombinant chromosomes or the recombinant loci when the alleles are exchanged with each other in non-sister chromatids.